All right, well, welcome everyone. My name is Lynn Mullen, and I'm a senior scientist with QIAGEN's Bioinformatics. And today I'm going to show you the analysis match tool, which is one of the newest and most powerful of IPA's features. It uses the power of IPA's causal analytics to compare your data with all of your IPA analyses, plus a massive set of publicly available data sets. And this can provide you with a unique option for making biological discoveries. This webinar is suitable for new users of IPA and also those of you who are experienced and you just want to learn more about this feature. It's scheduled to last for 45 minutes, and I'm going to spend about 35 minutes presenting and then open it up for questions for the remaining 10 minutes. And this is just a note that the software that we're presenting today's webinar is for research purposes only. And this is the last webinar in our series this month. So we started with an introduction to IPA, and then we followed it with a webinar about how you can format and upload data sets into the program. And in last week's webinar, I showed you how to interpret the results of an expression analysis, and it included a very brief introduction to the analysis match feature. And note that if you've missed any of these webinars, you can always watch recordings of them available on our website, and I'll show you the link to that website at the end of the presentation. But first, I'd like to provide you with a short overview about the webinar. I'm going to start by giving you an introduction to the analysis match feature, how the publicly available data sets were processed for comparisons, and also which type of IPA analyses are being compared. I'm next going to describe the data set that we're going to use in our live demonstration, and then during that demo, I'm going to tell you more about how the matches between the analyses are performed. And then after a short summary, I'll have some time to answer your questions. Okay, so what if you could identify other analyses that look like your own? Ones that had similar upstream regulators or activated canonical pathways and more. Or analyses that show very different biological characteristics than your own. That's the goal of the new analysis match feature. Now, before this feature was introduced, you could compare up to 20 different analyses using the comparison analysis tool in IPA. And now, the analysis match feature will automatically look for similarities and differences not only with all of your own analyses, but also with about 8,100 publicly available human, mouse, and rat data sets. So now you can feel more confident about your results. So for example, you might find that your results and other studies that examine the same disease show similar patterns of upregulation of key biomarker genes. You can find unexpected insights into biological mechanisms that are shared between different studies. So for example, you might find that your study and others that are examining ostensibly unrelated diseases all see an increase in fatty acid-related processes. And so that might be a clue to their etiology. And then analyses that are different from your own, so in other words, they could be considered as anti-matches, might provide you some key insights. So for example, let's say that you've given mice a new psoriasis treatment, and you perform the analysis match, and you find that the upstream regulators in your analysis show opposite activity patterns compared to rheumatoid arthritis samples. So maybe this drug that you're using for psoriasis could be used to treat arthritis. And I just want to make a really quick note here that the analysis match feature does require an additional license on top of IPA's Advanced Analytics license. And now I'd like to tell you about how these thousands of data sets and analyses were constructed for use in IPA. So these data sets were first downloaded from databases such as SRA and GEO and the Cancer Genome Atlas, among others. And the data are from microarray and RNA-seq experiments, and they were processed by software from QIAGEN's recently acquired OmicSoft. And then the curated data were put into what are called lands. And then we took the data from the lands and created comparison data sets. So for example, we could take ratios between experimental treatment groups versus non-treatment groups. And then we went ahead and uploaded those data sets into IPA for analysis. And then both the analyses and data sets are located in a special repository in the program. So the analysis match feature allows you to leverage OmicSoft's strength of curation of data together with the knowledge base in IPA and the analytical tools that it uses to confirm ideas, or you can gain some unexpected insights. Now we have 2 of OmicSoft land analyses available for this feature. So they are disease land, which contains information from human, mouse, and rat, and oncoland, which is a land that focuses only on cancers. So this land has information from OncoGeo, TCGA, and also 3 other categories for information from metastatic and hematological and pediatric cancers. Now, for both lands, we have a number of different tissues and diseases and microRNA and RNA-seq data sets available, and this number grows with every quarterly release. Now, there's several main categories of experimental types within these analyses in the lands. So these are classified as treatments versus controls. We have diseases versus normal phenotypes, as well as comparisons where you can directly compare different treatments, diseases, cell types, and more. And then we also have data sets that fall in, that if they don't fall into any of these categories, we put them into something called the other comparisons. So for example, data that from a study looked at patients that were in remission from a disease versus those that were recently diagnosed. Now, for each analysis, we create biological signatures using 4 main types of results from IPA. So we look at canonical pathways, upstream regulators, causal networks, so in other words, master upstream regulators, and diseases and functions. And I'm going to call these 4 types of results entities for the rest of the program, or presentation, excuse me. Now, for those of you who attended the data formatting and upload webinar 2 weeks ago, you might recall that I mentioned that you're going to want to analyze the most significant molecules in your data set rather than all of the molecules in the data set. So one way you can do this is when you're in IPA, you can set some cutoffs. So for example, you can use statistical values like false discovery rates or expression or phosphorylation values. So as you can imagine, with 8,100 different experiments, that there's going to be a lot of variation in the number of molecules that are truly significant within each study. So we therefore aim to standardize how many molecules would be able to be analyzed. So these are called analysis-ready molecules. So we decided to aim for 1,000 analysis-ready molecules per observation. So I'm going to illustrate this process by which we identify the most significant genes using an example from our OmicSoft dataset. And this was from a study that examined the response of breast cancer cells to hypoxia. And here's just a little cartoon of a gradient that represents the entire data set. So the green represents varying levels of downregulated genes, and the red represents upregulated genes. This study in particular used an Affymetrix array that had over 54,000 probes. Now, our first step in the process is to filter the data sets by applying a p-value cutoff of less than 0.01. And after we apply this p-value, if fewer than 50 genes pass the cutoff, then we just discard the data set and don't put it into IPA. If we have anywhere from 50 to 1,000 different genes in the data set, we do submit it for an analysis and stop right there. Now here we have over 2,000 identifiers. So we need to apply some further criteria to find the most significant genes to get it down to that 1,000 number. So rather than setting just a standard fold change or other expression measurement cutoff, we decided to look at each data set individually and determine the proportion of up or down regulated genes. So in this example, we have a lot more upregulated genes than downregulated genes in this data set. So we can then set some cutoffs to reflect these proportions. And when we're doing this, we end up with about 711 upregulated genes, which range from a fold change expression value of about 0.5 to 2.6, and 291 downregulated genes. So here we have a little bit different cutoffs just to reflect that proportion, that balance. And just to note, sometimes what you're going to see when you're doing the analysis is that, oops, what's going on here? Oh, sorry. Sometimes you're going to see 1,000 genes. Even if you try to aim for 1,000, sometimes what gets uploaded is fewer than 1,000. As I mentioned before, sometimes you may get to 1,000 and it's close to 1,000. You might say, okay, let's call it a day. But in some cases, you've gone to about 1,000, and the reason you see fewer is because some of those identifiers were duplicates for the same gene, and IPA only picks one identifier per gene, or they're merged into orthologs when they're uploaded into IPA. And now I just want to introduce you to the example that we're going to use to demonstrate the analysis match feature. So this is the same data set that we've been using in the last 2 webinars. And it's from a study that examined different cell lines of melanoma. And melanoma is considered to be metastatic melanoma is considered to be the deadliest form of skin cancer, and it develops from melanocytes, which are the cells that create pigment in the skin. The primary cause is thought to be due to DNA damage to skin cells that are caused by exposure to ultraviolet radiation, and that can be from tanning booths or from the sun. Now, it usually occurs in the skin, but sometimes it might be found in other organs like the eyes or intestines or mouth and more. And it tends to most often spread to the liver, lungs, bones, and brain. Now, melanoma can be treated successfully if it's detected early, but this disease can metastasize very quickly, and it can be difficult to treat at the later stages of the cancer. In fact, the median 5-year survival is only 15 to 20%. So in this study, the researchers measured differential gene expression using a whole genome microarray to characterize the different melanoma skin cells. And we decided to just pick one of these cell lines. So it's a metastatic melanoma line that is compared to a cell line that was generated from normal skin. Okay, and with that, I now want to go into IPA. All right. So I just want to give you a really quick overview of our welcome screen. So here in the middle, you have our quick start menu. So we have a place where you can look at different tutorials or videos. And here is a shortcut so you can use these analysis wizards to get started with your different analyses. At the top, we have a search engine, so you can search for different genes and diseases, chemicals, etc. And then over here in the file, we have the ability here to upload your data set or to begin different analyses. And then you can also use many of the same options here with the new button. And under here, we have the project manager. So the My Projects folder is where your analyses and your data sets are stored. And then we have this library section. So when you have the OmicSoft or the Analysis Match license, you'll see this library here for OmicSoft. And you can see as I click on these plus signs to expand, you can see the 2 oncolands, oncoland and disease land. And as I keep clicking, you can see the different subcategories. And finally, you can go ahead and you can dig down and actually access those dataset files and analyses for yourself. All right, so I've already opened the analysis for the metastatic melanoma, and I've minimized it. So I'm going to go ahead and maximize it right here. Now, in last week, I showed you the results of this expression analysis. So we have results showing different enriched canonical pathways and upstream regulators, diseases, and more. And if you'd like to watch that webinar again, I just want to remind you that we do have a recording available of that on our website. Now I want to take a look here at the analysis match feature. And here it is. So by default, Analysis Match is going to provide matches to the OmicSoft datasets, but also to all of your own analyses you've already performed that are in your My Projects folder. Today, just to keep things a little simple, I've just filtered this so you only see the OmicSoft data. But when you click on this little filter icon, you can see this is pretty much what you just saw in the My Project. So project manager, excuse me. So I could go ahead and I can choose all of my projects or just a few, or I could go ahead and just pick, if I'm only interested in pediatric matches, for example, I could go ahead and do that there. And then if I want to go ahead and include or exclude certain analyses, I can filter for that using these different wildcard searches right there and just type in that information there. Okay. Now, before I describe what's in the table, I do want to point out that there are many options here that do remain hidden. because there's quite a lot of information we have here, but we have even more. So when you go into the customized table, you can see all of the different options that you could add that could be separate columns within this table. So if you want to sort, for example, by ethnicity or gender or smoking status, for example, when you click on these little check boxes, those columns will appear in the table. One of the benefits of adding this information when you're uploading your data set is that you can more easily filter for matches here in this Analysis Manage feature. So in the previous webinar in this month's series, I did talk about how you can associate different metadata during that upload process. And you can view any of the metadata that you have associated with your analysis in the Summary tab. So here's the experimental metadata. And here you can see that I had added metastatic melanoma as a case cell line. All right. Okay, so let's go back into that tab right there. Now, the default columns that describe the metadata are the disease state. We have the type of tissue that's used in the experiment, the comparison category. So this tells us information about what comparisons were made. And I mentioned that on the other side. So for example, here we have disease 1 versus disease 2. And then the comparison contrast gives us even more specifics about the type of comparison. And so we can go, expand this, and you can see, for example, this disease 1 versus disease 2 is actually a comparison of ulcerative colitis versus Crohn's disease. And then finally, we have the comparison ID. So this is the source of the data. So you can see we have these gene expression omnibus series numbers listed here. So what you can do is you find something that's really interesting and you want to learn more about it, you can just type in the number into your search engine and it'll take you straight to where you can find that paper and also the sample data. And then we also have after that number, we have a .gpl number, and that is the microarray that was used, if it was from a microarray, that is. The remaining columns here consist of the ranks of the matching scores. And we score the results in 2 different ways. So the first 5 columns here show you the z-scores, and then the final columns show you the p-values. And for each one of these, we look at the entities. So just as a reminder, our canonical pathways, or CP, UR is upstream regulators, CN is the causal network, the master upstream regulators, and DE stands for downstream expression. So in other words, the diseases and functions. So now what I'd like to do is I want to go back to my slides and tell you more about how these z-scores and the p-values were calculated. So let's go ahead and we're going to start with the z-score calculation. Now, I'm going to be talking about 2 different z-scores today. So the first is the z-score that's calculated in a core analysis, an expression analysis, phosphorylation analysis, etc. And I talked about this in more detail in last week's webinar. So that z-score aims to assess the strength of a prediction of activation or inhibition of an entity. For the analysis match, we're going to use those z-scores to generate a new z-score, and that's used to determine the significance of a match between 2 analyses. So first, what we do here is we match the analyses. So we're going to use an example of upstream regulators. And now we have a new gradient. So here the color is blue stands for all of the upstream regulators in your analysis that are inhibited, and orange represents those that are activated. And just as a reminder, when we're looking at those activity predictions, the statistically significant z-scores have an absolute value of 2 or greater. And those are represented by the darkest colorings in this gradient. So for this step, when we're trying to choose the most significant upstream regulators, we use the absolute value of a z-score greater than 2 and also a p-value of less than 0.05. And what we do is we take the n topmost entities, so in other words, those that have the strongest statistical support for predictions of activation, and the n bottommost entities, so those that have the strongest support for predictions of inhibition. And currently, n is defined as 50 for all entities except for canonical pathways, for which n is 10. So in other words, in this example, we're considering the 50 upstream regulators that have the highest z-scores and the 50 most significantly inhibited regulators, so the most negative z-scores. So you can think of this process as creating a signature for the upstream regulators in our analysis. What we do then is we query the signature against all of the analyses, and we calculate the z-score to determine if the match is strong or weak. So here I have 4 different illustrations of what you might potentially see. So here our gradient again represents our analysis, and then all of these different lines here represents an upstream regulator that's a match from another analysis. And the first example here shows a strong positive match. So you can see there's many lines within this significant 50 boxes here. So this indicates there's a large number of matching upstream regulators in that query data set. And also we can see their activation patterns are pretty similar. So most of the upstream regulators that we are showing as activated in our analysis are also activated in the other analysis, and likewise for the inhibited upstream regulators. The next 2 gradients here represent 2 types of weaker matches. So here there are fewer lines, so there's fewer matches overall, and their activity patterns aren't always as consistent. So some are upright or activated when ours are inhibited, for example. And then the final gradient here represents a match, a strong anti-match. So many of the significant upstream regulators in our data set are also present in this other analysis that we've queried, but they all show opposite activity patterns. So this is an example of a data set that has an opposite biological signature to our own. And I just want to note, though, that each gene counts equally within the top and the bottom 50 upstream regulators. So once you have some biological attribute or molecule that's in that top 50 or in that bottom 50 list for an entity, then the magnitude of their z-scores and their p-values are not considered when we're trying to make a match. Okay, so now what I want to do is let's take a look and see how the analysis match z-score is now calculated after we've tried to make a match. So again, we're looking at using the z-score, and it's going to be a measure of the matching of the molecules or those biological attributes that are associated with some entity between the analyses. So we're assuming that the pattern is created from 2 sets of entities where the sign of the matching entities is random. So here, we're using that absolute value of 2. So a z-score that's greater than 2 or less than -2 is statistically significant. So over here, I have a very simple little cartoon that shows 10 different upstream regulators. So each upstream regulator is represented by a circle. And we're using the same color coding for activation of orange for activated upstream regulators and blue for inhibited. And so what we do is we perform the match, and then we find that in our matching analysis, we see that these 5 upstream regulators are showing a perfect match. So here, these are represented by these plus signs, and we also see that with the H, I, and J are also perfect matches. We can see here that G is a match, but it shows an opposite activity pattern, so it's represented here with a minus sign. And then finally, F is in our data set, but not in the matching data set, and vice versa for K. It's in the other analysis, but not in our analysis, so it doesn't get considered in the z-score as a match or an anti-match. Okay. So from this information, we use the following equation to calculate that match z-score. And so here we have 8 matches and 1 anti-match, and we divide by the total number of molecules that match, and then we get a value of 2.33, and this is greater than our cutoff value of 2, and so therefore we make conclude the analyses do significantly match. Now, we also wanted to normalize the z-scores so that we can compare them across different data sets. And we also wanted to simplify how we reported these results. So instead of reporting the actual z-score in that table that I showed you in IPA, we instead converted them into percentages. So to do this, we used the same z-score formula, and we calculated what the z-score would be if we were comparing our analyses with a perfect match. And it's essentially what it comes out to using that same z-score equation. It is essentially the same as the square root of the number of entities that we're trying to match. And then what we do is we divide the actual raw z-score by this maximum value. So in the example that I showed you from the previous slide, our raw score was a 2.33. And then if we divide this value, the 2.33, by our perfect z-score match, we get a match of z-score similarity of 74%. Okay, so now what I'd like to do is talk about the p-value of overlap here. So this is calculated using the Rechtel-Fisher's exact test. And again, it is different from the p-value of overlap that's calculated with a core analysis. And here we're using this test to calculate the probability that the number of overlapping molecules between the two sets are due to chance alone. And the null hypothesis here is that the sets are independent. So for analysis match, the p-value is going to depend upon the size of the set of the two entities, and here we cap it at 100. We look at the size of the overlap between the two entities and the universe of all entities across all analyses. And then finally, what we do is we calculate an overall z-score and an overall p-value. And the overall z-score is pretty simple. It's just the average of the z-score percentages among the four entities. And then the overall p-value, it's also a percent similarity. You might notice in this little screenshot here, though, that the actual p-values for each individual entity is not reported. Instead, the actual p-value is reported, but the percentage is not reported. And the reason we decided to do this is because the percentages turned out to be harder to interpret. Now, to get that overall p-value, we take the negative log of the p-value for each entity, and the maximum value we used was 50. So if you have a negative log of a p-value that's greater than 50, it's just set to 50. And then we normalize each of the p-values against a perfect match to get the percentages for each entity. And like the z-score, we just take the average for all four entities. All right, let's go back into IPA and we'll take a closer look at those z-scores and p-values and their matches. Now, before we look at the scores of the matches in more detail, I just want to tell you about how you can interpret the names of the matching analyses. So the analysis name is structured so that it starts with test and a number. So this terminology refers to the specific comparison that was run from the public data set. So for example, let's consider a study that had 5 experimental groups. If you see a match that says test 2, it would mean that would have been the second experimental group comparison in that study. The next part of the name tells us the disease state of the case. So in other words, the experimental group. So for example, here, B-cell lymphoma. And then in brackets, we see the tissue from which the RNA or the DNA was extracted. Now, if you had a treatment that was given to a particular experimental group, then it would be listed at the very end of the name. Sometimes you're going to see none at the end. So what this means is that the experiment had treatments, but this particular group did not have the treatment. And then you'll see NA. So what this means is that none of the experimental groups in the study were given a treatment at all. And one final thing, you might see the term normal control. It doesn't mean that this analysis represents just a control sample. Instead, what it means is that the starting tissue came from normal tissue, and it was induced to have some sort of effect. So for example, it could have been treated with some sort of chemical to stimulate rheumatoid arthritis, for example. So just kind of glancing at these results, we can see by eye we have the B-cell lymphoma as the highest match, a number of lung cancers, and a number of other diseases. And sometimes you're going to see results that surprise you, but it's really important to not discount them offhand. So you're going to want to keep in mind that these matches could give you a clue that there are some diseases or phenotypes that have similar mechanisms of action that were previously unknown. And in particular, your deeper knowledge of your experimental background and the conditions could really help you make sense of the matches. So for example, I had mentioned earlier that metastatic melanoma doesn't just affect the skin. So it can also be found in the intestines. And we can see there are some intestinal disorders like ulcerative colitis and Crohn's disease that do show up as matches here. And also, the brain is one of the most common places for the cancer to metastasize, as well as the lung. So we do see evidence of matches that are to these types of organs here. So you could perhaps say that, let's say if I develop a treatment for metastatic melanoma, maybe it would help in cases where you have, say, Crohn's disease or ulcerative colitis. Now, one important thing that you're going to want to do, especially when you're looking at matches that you're not expecting, is to look up the paper that it's been reported from, because that's going to give you a lot of information as to exactly what was done and what this means. Okay. All right. So one powerful thing you can do also is to filter this information to get to a smaller set of results. So let's say that I'm only interested in seeing matches from humans. So I could pick Oncoland, and let's also pick hematology and human disease. And I'm going to click apply. And then let's get rid of some of these lower overall z-scores. I'm going to choose 45% match, and I'll also do a p-value match of 45%. Okay. So it doesn't really, we can't really give a recommendation for a percentage cutoff for everyone's analyses, because your analyses matches and your needs are going to be really different. So you just have to kind of play around with the values that make sense for you. Now, the z-score, the overall z-scores runs from 100% positive to 100% negative. And we chose the scale because it can be really powerful to see not just what data sets are like yours, but also find ones that show very different results. And those different results, those anti-matches, are shown with this light blue color. So again, these are the analyses that show very different biological signatures from our analysis. And as you're scanning by eye, you might notice that there's a number of different diseases and infectious diseases like the flu and the common cold that show up here. I went ahead and I read a number of these different papers before the webinar, and I found that there was a couple of or tissues that were stimulated with some pro-inflammatory agents. So here we're finding that metastatic melanoma has a biological signature that is quite different from infections and inflammatory responses. And this information for these matches can be really powerful if you find some treatment that's having an effect on a disease. So for example, it would be really cool in this case to find an anti-match from a study where cancer cells or tissues were treated with some drug, because if it shows up as an anti-match, that means that maybe that treatment could be effective against our disease. So if you want to go into more details about the core analysis that was run for a match, you can always just click on the name and it will take you straight to the core analysis that we ran in IPA. So once you find those matches, you filtered it down to what seems to be most interesting to you. You might be interested in finding which specific entities are driving these patterns of similarities or differences among the data sets. So for example, when we look at these different entities, we can see the z-score similarity is pretty high for upstream regulators compared to the other entities. So what are the upstream regulators involved? Why are we seeing these matches show up? So what you can do here is you can select as many as you like. So I can go ahead and just select all of these, and you can click on View as Heat Map. So I've actually already opened that, here, and this is the window that you get when you first open that up. So this is structured so that each column is one of the analyses, and the our analysis is highlighted in pink. And each row represents one of the entities. And you can see also there's a dendrogram on the left side of the screen. And this shows you which of these entities cluster together. So these clusters can reveal some interesting similarities among the different entities. So for example, this cluster of different causal networks and upstream regulators are likely interacting with each other. And when you hover your mouse over the area of the dendrogram of interest, you probably notice that it's turning red. So if I went and clicked on it, just one click, you can see now it's blue and it highlights the names of the entities of interest. You can also choose this cluster columns option, and what this does is it gives you the option to view the heat map as a function of clustering similar analyses together. So when we do that, you can now see that these sets of analyses are quite similar to our own, and the ones that are most dissimilar are over here. Now in IPA, you probably have noticed, well, gosh, there's drugs located in here, but this was gene expression or RNA-seq data sets. So I mentioned in our last webinar that we do consider upstream regulators to be any type of molecule that has some sort of downstream effect. But if you're not interested in these, you only want to look, say, at the genes, you can go to the filter option. There's a number of different filtering that you can do. I'm going to just choose the molecule filter, the genes, RNAs, and proteins, and it just automatically recalculates everything for you. Okay. Another really cool feature that you can do is, let's say, oh, it's really hard to see, is that a significant Z-score or not? You can tell if you hover your mouse, but it's kind of hard to do this for everything. One thing you can do is you can set an insignificance threshold. So if I use that statistical cutoff of the absolute value of 2 for our Z-scores and hit apply, then I get these little dots here. So I have a really quick and easy way of finding out which ones of these Z-scores are actually not significant with that, my cutoff. And you can also view the metadata for each matching analysis. So all you have to do is just click on the name. And over here we can see all the metadata. So this includes information like the different tissues and also the fold change cutoffs that were applied and more. And if you want to gain even more information about the similarities and differences, what thing you can do is you can choose up to 19 of the different matches, so for a total of 20, and then just choose View Comparison, and that will open up a comparison analysis. And you can also do the same thing in the expression analysis. There's a View Comparison option right here. I'll just choose some right there. Okay, and with this, I'd like to end the live demo of IPA, and I just want to go back to my slides and just give you a summary of what we've talked about today. So the Analysis Match tool allows you to compare your analysis to the analyses from thousands of publicly available data sets, as well as all of the data sets and analyses that you've performed. So you can obtain and critically examine hypotheses by just finding the analyses that have similar or opposite biological patterns to your own. It allows you to build confidence in your results by identifying matches that have shared biological signatures across a variety of different disease states and phenotypes, drug treatments, and more. Unexpected matches can give you greater insight into the biology shared among disparate diseases and tissues, etc. And anti-matches can give you powerful information about which molecular mechanisms and biological processes are quite different from your own study. And you can also develop greater insights into your own analysis by identifying the specific pathways and regulators and downstream attributes that are driving similar or opposite phenotypes. All right, so this completes the webinar. I hope you've gained a deeper understanding of the Analysis Match feature in IPA and how it can help you with your research. We're going to send you a recording of this webinar in the next day or so, and it will also be uploaded onto our website. So here's the link to that website. I refer to it sometimes as the TV channel. So here we have previously recorded webinars. This is just a small example of what we have. And we also have some video tutorials there that go into even further detail about these topics. And if you'd ever like to contact us, you can call us at any of these phone numbers or email us at this email address. If you're interested in finding out more information about the Analysis Match license, you can contact your QIAGEN Bioinformatics account manager or just send us an email at bioinformaticssales@qiagen.com. And if you want more information about IPA and other software and databases, you can find that at QIAGEN.com or QIAGENBioinformatics.com. And here again is our TV channel link. All right, I'd like to thank you very much for watching, and I'm ready to open up for questions. So if you have any questions, please feel free to type them into the question box if you haven't already done so. And I just want to make that note that if I'm unable to answer your questions in the time we have remaining, I will personally email you with a response by tomorrow. All right, thank you very much for your attention.
QIAGEN.
Sample to insight.